Hello, welcome to Selectus, and thank you for joining us for this episode 2. I'm Laurent Poirot, Senior Vice President for Immunology. Selectus is currently involved in 7 clinical trials, representing 4 partnered products and 3 wholly owned products. This achievement represents years of work and expertise developed around 4 pillars of technology. First, protein engineering, representing more than 21 years of experience on several generations of nucleases, as well as CAR engineering. Second, vectorization using viral vectors. Third, electroporation with our proprietary device and buffers. And of course, cell and gene engineering that we transformed into a robust manufacturing process. What brought us to the clinic is now allowing the deployment of the next generation of CAR-T products. Therefore, I'm happy to share with you an overview of what's coming up next for us in the clinic.
Hello, I'm Beatrice, a team leader in the Immuno-Oncology division at Cellectis, driving CAR-T cell development. Today, I'm honored to present Cellectis' first allogeneic dual CAR-T cell product candidate for the treatment of B-cell malignancies that we expect that will be coming soon into clinical development. First, I would like to dedicate some time to explain the function of the immune synapse, a highly organized structure that is formed between the CAR-T cell and the target tumor cell. On the top figure, we see a B-cell tumor in blue and a CAR-T cell where the CAR is labeled in red. At the connection of the tumor cell and the CAR-T cell, the CAR form clusters as highlighted by the accumulated red, and thereby engages in the immune synapse, which is crucial for CAR-T cell activity. How does this work? The tumor cells have several antigens surrounding the cell. The CAR-T cell also expresses many CAR molecules around the surface. Now, when the CAR-T cell encounters the tumor cell, CAR molecules cluster at the junction with the tumor cell and engage in the immune synapse. It is then when the CAR-T cells get activated and start a signaling cascade that leads to the release of cytokines and deadly proteins such as granzyme or perforin that attack the tumor cells and kill them. And that is how the CAR-T cells achieve their therapeutic effect. Antigen downregulation and loss are frequent reasons for relapse in patients treated with CAR-T cell therapies. Now, what does this look like? When tumor cells present an abundance of the antigen, a strong immune synapse is formed, leading to strong killing and a good therapeutic outcome. However, when the antigen is downregulated, there is still some binding and killing, but it won't be as strong or persistent, and CAR-T cells can exhaust. The main issue arises when the antigen is downregulated to insufficient levels to facilitate a strong immune synapse, and the signaling cascade required for efficient tumor killing is impaired. Worst case scenario, the antigen is lost and the CAR-T cell cannot recognize the tumor cells and kill them. This phenomenon is called as immune escape and results in failed therapies. We believe in dual CAR therapies as a solution to overcome immune escape and provide better therapeutic outcomes. Let me explain how. In a dual CAR therapy, we have 2 CARs that recognize 2 different antigens or targets in the tumor cell. Therefore, in this case, more antigen is available to facilitate a tie formation between the immune synapse and lead to a robust killing. A benefit is that even if there are lower quantities of both of the targets in the tumor cell, since the dual CAR-T cell carry 2 different CARs, they can form a strong immune synapse and provide robust killing. Another benefit of dual CARs is that they are able to initiate the killing cascade in the presence of only one of the antigens, or the other. Therefore, we believe that targeting 2 antigens has the potential to reach a large patient population and to mitigate the risk associated with antigen level downregulation or loss. And using this strategy, we expect to see successful therapeutic outcomes. The restricted expression of B-cell surface markers to the B-cell lineage make them very appealing candidates for CAR T-cell therapies. While B-cells present several attractive targets for immunotherapies, CD19 has gained the focus of attention worldwide as it was the benchmark for CAR T-cell therapies, and it became the first one to be approved by the authorities. And yes, CD19 is a well-known B-cell surface marker and an attractive target expressed in a large variety of B-cell malignancies, as we can see here. However, we believe that CD20 and CD22 are also excellent targets for CAR T-cell therapies. They are well-validated B-cell targets, and as we can appreciate in this figure, both CD20 and CD22 are similarly expressed in a large variety of B-cell malignancies, and they are safe targets whose expression is limited to the B-cell lineage. I'm thrilled to introduce you to UCART 2022, the first therapy of its kind in the allogeneic CAR T-cell space that targets both CD20 and CD22 simultaneously. UCART 2022 targets two validated targets in B-cell malignancies, and actually rituximab, an antibody targeting CD20, is the standard of care for several types of non-Hodgkin lymphoma. As this dual CAR T targets two validated and safe antigens, we believe that this innovative therapy will prevent immune escape due to antigen loss and will strengthen the immune synapse, leading to a robust killing, and this will translate into positive therapeutic outcomes. We do not anticipate additional risk since UCART 2022 carries the same talent-mediated gene modifications that our products that are already in the clinic carry, UCART 123, UCART 22. And these talent-mediated modifications target the TRAC locus to prevent graft-versus-host disease and CD52 to favor CAR T-cell persistence upon co-treatment with anti-CD52 antibody. UCART 2022 has the potential to serve a large patient population and considerable market access since it can be used to treat both B-cell acute lymphoblastic leukemia and non-Hodgkin's lymphoma. Altogether, the incidence rates of these two diseases add up to more than 200,000 patients per year, and CD22 is expressed in over 90% of B-cell acute lymphoblastic leukemia patients, and CD20 is expressed in over 90% of non-Hodgkin lymphoma patients. Thus, these targets cover a vast spectrum of B-cell malignancies and a large percentage of the patient population. And now, let me show you some of the exciting in vitro data that we have generated with UCART 2022. Here, we can see how UCART 22 cells are efficient killers of tumors expressing both antigens, CD20 and CD22, or only one of them, but fail to kill tumors that express only CD20 or that do not express any of the antigens. What makes UCART 2022 very attractive is that when we treat the same tumor cells with the dual CAR, we do not only achieve robust activity, but also restore the activity against CD22 negative cells. We have also monitored the activity of UCART 2022 cells over time by exposing them numerous times to tumor cells expressing a combination of the antigens, either both, one or the other. And as we can see from this figure, the powerful activity of UCART 2022 was persistent for over 2 weeks, regardless of which target or which target combination was expressed. Therefore, we believe that this robust and prolonged activity will translate into efficient and safe therapies with the potential to target a large patient population presenting a variety of CD20 and CD22 antigen combinations, and also to address relapses due to antigen modulation. We also generated in vivo data that are consistent with our in vitro data. So here, we generated an innovative mouse model where we injected 3 different antigen combinations in 3 different flanks. In one of them, we injected double positive cells, CD20, CD22. In the other one, we injected CD20 only positive cells. And in the third one, CD22 only positive cells. This allowed us to follow the tumor progression in 3 different tumors upon injection of UCART 2022 in parallel. Animals treated with UCART 22 only succumbed earlier to the disease because these animals also carry tumors that do not express CD22, and those tumors were able to grow. For UCART 2022, the dual CAR, we observed a dose response effect where animals treated with 8 million cells presented a longer survival time. When we now focus on the progression of the double positive tumor cells CD20, CD22, we observed that already a low dose of 3 million cells of the dual CAR can elicit a strong tumor control, contributing to the long-term survival that we observed. In a disseminated lymphoma preclinical model, we used double positive cells, CD20 positive, CD22 positive, and we were thrilled that already treating them with only a low dose of 1 million dual CAR T cells, we were able to achieve a very robust anti-tumor response and significant long-term survival. What was also very exciting is that when we examined the bone marrow of these animals treated with the dual CAR in the disseminated model of lymphoma, we observed that the CAR T cells persisted for over 100 days, consistent with sustained therapeutic activity for a long time. And with this, I would like to conclude with the highlights of the first dual CAR T cell candidate product developed at Cellectis. It is the first of its kind in the allogeneic CAR T cell space, and we have seen that UCART 2022 provides robust and strong in vitro and in vivo preclinical results. UCART 2022 has the potential to reach a large patient population that encompasses both non-Hodgkin lymphoma and B-cell acute lymphoblastic leukemia patients with an incidence of over 200,000 cases per year, and can target also CD19 and CD22 relapses. We already have done this in the past, meaning that we have the manufacturing and regulatory tools to bring this new UCART 2022 product candidate soon to the clinic. And with this, I would like to thank you all for your attention.
Hi, I'm Cécile, and I'm project leader at Cellectis. Today, I'm going to present you the work that we have done concerning the development of the UCART meso product. On this pie chart representing the number of CAR T trials per cancer type, we can clearly see that, to date, the most clinically advanced indications for CAR T therapies are hematological malignancies. However, hematological malignancies represent only a minor fraction of the total cancer case. There is thus a huge medical need for solid tumors. This is in this context that Cellectis decided to start the development of a UCART product for the treatment of mesothelin-expressing cancers. Mesothelin is a very attractive target for solid tumors. It is highly expressed on different cancers such as ovarian cancers, mesothelioma, pancreatic cancers. In addition, mesothelin is one of the most studied targets for solid tumor treatment, and promising preliminary clinical results were obtained with the mesothelin-targeted agents. Different factors present in the tumor microenvironment can impair CAR T cells' activity. This is, for instance, the case of TGF-beta. In a TGF-beta deprived tumor microenvironment, the recognition of the solid tumor cells by the CAR T cells induces its activation. Whereas in a TGF-beta rich tumor microenvironment, the binding of the TGF-beta to its receptor, expressed at the CAR T cell surface, induces the inhibition of CAR T cells activation. We have shown that CAR T cells exposed to tumor antigen in the presence of TGF-beta display decreased expression of IL-2 receptor, suggesting that these cells have impaired proliferation and survival capacities. One possibility to counteract the inhibitory effect of TGF-beta on CAR T cells is to knock out the TGF-beta R2 gene. TGF-beta HER2 knockout CAR T cells can be activated in the absence of TGF-beta, but also in the presence of TGF-beta. We have shown that TGF-beta HER2 knockout CAR T cells exposed to the tumor antigen in the presence of TGF-beta display restored expression of IL-2 receptor, suggesting that these cells have normal survival and proliferation capacities. Having shown that blocking the TGF-beta signaling pathway in CAR T cells could improve CAR T cells activity, we decided to generate meso-CAR T cells knockout for the TGF-beta HER2 gene and to analyze the capacity of these cells to kill H226 cells, a mesothelioma cell line expressing mesothelin and that is also able to produce TGF-beta. We first did an in vitro experiment in which we analyzed the lysis of H226 cells 24 hours post-initiation of a co-culture with CAR T cells. We observed that a very efficient killing of H226 cells was happening and that this killing was proportional to the dose of CAR T cells. We then decided to evaluate these cells in a mouse model of mesothelioma. To generate this model, we used immunodeficient mice. The mice received intrapleural injection of H226 cells, and 13 days later, the tumor burden was analyzed by bioluminescence imaging. As you can see, the tumor burden was pretty high in all the animals. 1 day later, the mice received IV injection of vehicle or CAR T cells. And then, at different time points, the tumor burden was analyzed to assess the effect of the different treatments. We have shown that the tumor of the mice that had received vehicle had grown over time. By contrast, the tumor of the mice that had received 1 million CAR T cells had regressed over time. The tumors were almost undetectable at the dose of 3 million CAR T cells. Altogether, these results clearly demonstrate that TGF-beta HER2 knockout CAR T cells display very potent anti-tumor activity in vivo. The UCART mesoproduct that we are currently developing for the treatment of mesothelin-expressing cancers has the following characteristics. It corresponds to a T cells that express a chimeric antigen receptor directed against mesothelin at its surface. The chimeric antigen receptor being vectorized using an RLV vector. The T cells are also knockout for the TCR alpha and the CD52 genes to respectively prevent graft versus host disease and improve the persistence of allogeneic T cells in the presence of alemtuzumab. The T cells are also knockout for the TGF-beta HER2 gene to protect them from the inhibitory effect of TGF-beta. The take-home messages from this presentation are that TALEN gene editing unlocks CAR T cells activation in TGF-beta enriched environment, that TGF-beta HER2 edited meso-CAR T cells shows potent anti-tumor activity, targeting TGF-beta signaling could be beneficial for multiple solid tumors. Thank you very much.
Following mesothelin in indications such as pancreatic cancer, the next target we're pursuing for solid tumors is MUC1. One important challenge in treating solid tumors is that most molecules expressed in cancer cells can also be present in some healthy tissues. And when targeting those, these can lead to serious adverse events away from the tumor. MUC1 is of high interest in this regard for several reasons. First, although MUC1 is expressed in normal epithelia, it is only present on the apical side of these cells, facing the external space, where it is not detectable by CAR T cells that are in circulation inside the organism. In tumors, the polarity of distribution of MUC1 is lost, and MUC1 is present all around the cells. There, CAR T cells should easily detect it. Second, normal MUC1 is heavily glycosylated, meaning that extended networks of sugar molecules cover this protein and hide it from T cells. This is what creates the mucus at the surface of mucosa. In cancer, glycosylation of MUC1 is greatly reduced. Normal MUC1 and cancer MUC1 can really be seen as two different molecules. Our CAR is specifically designed to recognize MUC1 protein associated with short glycans. This property can be seen here. This signal indicates that our CAR is able to bind cancer cells, as shown in red, but is not able to bind normal cells that do express MUC1 in blue. This means that our CAR T cell should be able to bind and attack cancer cells, but should not be able to eliminate normal cells. This should be a therapy that shows good efficacy against cancer cells, but a good safety profile. Tumors are made of cancer cells, of course, but also networks of other cells and molecules that promote tumor growth. TGF-beta can be expressed by most of these cells. It is a great molecule to target to increase the efficacy of CAR T cells. We have achieved this blockade by either inactivating TGF-beta receptor on CAR T cells or by expressing a dominant negative version of this receptor that can act as a decoy. The beauty of TALEN gene editing is that it is precise, highly efficient, and specific. This allows multiplex engineering of the cells and the creation of networks of genes through synthetic biology. We therefore decided to go one step further and leverage the efficiency of TALEN gene editing to incorporate multiple functional attributes into the cells and increase the chances of therapeutic success. We therefore created a product where TCR is inactivated to prevent graft-versus-host disease, and a CAR targeting MUC1 is expressed using a recombinant lentiviral vector. A dominant negative TGF-beta receptor is also expressed using the same recombinant lentiviral vector. IL-12 is knocked in at the PD-1 locus, and HLA-E is knocked in at the B2M locus. Overall, we are combining 3 TALEN knockouts and 2 knock-ins. We already achieve high efficiency for this ambitious strategy. As an example of early engineering results shown here, we achieve close to 80% of TCR B2M double knockouts, very high inhibition of PD-1 expression, and over 60% of HLA-E expression. Overall, before any cell enrichment or depletion, greater than 30% of the cells are CAR positive, triple knockout, and knock-in for HLA-E. Let me show you what some of these attributes do for the cells. Allogenic persistence is an important property for allogeneic T cells. Inactivating B2M at the surface of CAR T cells renders them invisible to the patient's cytotoxic T cells. It should delay their rejection. As shown here in vitro, as we are increasing the number of alloreactive T cells mixed with our B2M negative cells, B2M knockout cells are getting enriched more than tenfold on average over B2M positive cells, indicating they are resisting rejection by alloreactive T cells. Inactivating B2M can nevertheless render T cells sensitive to NK depletion, as shown here in red, where B2M negative cells are close to 80% depleted when mixed with NK cells. In our cells, we are expressing HLA-E instead of B2M. HLA-E is an inhibitor of NK cells, and it confers resistance to NK cell depleted mediation, as shown in blue, where CAR T cells expressing HLA-E are indifferent to the presence or absence of NK cells. In our cells, substituting HLA-E for B2M should therefore confer resistance to both the patient's T cells and the patient's NK cells, therefore extending the therapeutic window in allogenic settings. We aim at further increases therapeutic potency of our cells with this second disruptive insertion. We knock out the inhibitory receptor PD-1 and insert IL-12 gene instead. This one-step manipulation serves several purposes. IL-12 is a potent immunostimulatory molecule. It can increase the therapeutic efficacy of CAR T cells themselves, but it can also attract pro-inflammatory cells in the tumor microenvironment. We believe this strategy can shift the tumor microenvironment from something that promotes tumor growth into something that promotes anti-tumor immunity. The great thing here is that we are making smart CAR T cells. PD-1 is only expressed once CAR T cells get activated. By inserting IL-12 at the PD-1 locus, it is only expressed at the tumor site. As shown here in vitro, when following IL-12 expression after 2 different tumor challenges, it follows exactly the kinetic of expression of the activation marker CD25. We published recently that this activation-dependent IL-12 expression increases the anti-tumor potency of CAR T cell in a lymphoma model. As shown here in vitro, when we follow the number of tumor cells after mixing them with CAR T cells, IL-12 expressing CAR T cells can more efficiently kill tumor cells, especially after the second tumor challenge. In vivo, although normal CAR T cells are able to provide some anti-tumor activity, as shown in red in this survival curve, only the group of mice injected with IL-12 expressing CAR T cells contain long-term survivors. We more recently evaluated this activation-dependent IL-12 expression in a MUC1 model. This graph represents the fraction of tumor cells versus CAR T cells within the tumor. When mice were either left untreated or treated with untransduced CAR T cells, there's no T cell infiltration within the tumor. Normal CAR T cells are able to infiltrate the tumor, but there's a balance between tumor cells and T cells, and there's really no tumor regression. The contrast with IL-12 expressing CAR T cells is striking. There, CAR T cells are able to proliferate within the tumor and cause tumor regression. This results in great anti-tumor activity, as shown here, on the left in a mammary fat pad tumor, and on the right in a subcutaneous tumor. To conclude, I'm very excited to share with you these latest results. MUC1 is a promising target to treat triple-negative breast cancer. In diseases where targeting TGF-beta signaling might not be enough, we can synergize strategies to increase potency. TALEN gene editing allows highly efficient, complex engineering where you can combine enhanced but controlled potency with allogenic persistence. Thank you very much.
Hello all, I'm Shipra, Team Leader in Innovation Immunology at Selectus, where we are working on the early stage of research and development of CAR T-centered strategies for curing cancer. Today, let me take you through the development of UCART FAP, our exciting new strategy for combating the problems posed by a solid tumor microenvironment against successful immunotherapy. So, when we speak of immunotherapy, especially in the context of solid tumors, what immediately comes to mind are checkpoint inhibitors. So, PD-1, PD-L1, CTLA-4 blockers, which restore to the patient's own immune cells the ability to mount an anti-tumor response. Checkpoint inhibitors have thus really revolutionized the landscape of cancer therapy, with clinical approval in more than 25 cancer indications and upwards of 4,000 clinical trials underway. Unfortunately, not everything is perfect. Checkpoint inhibitors are plagued by their own set of challenges. For example, while combination of these inhibitors are clinically advantageous, patients are often saddled with deleterious side effects. And then there are the notorious cold tumors, characterized by a microenvironment which is devoid of cytotoxic cells. And so, in a majority of patients, when there are no T cells in the tumor to get activated upon checkpoint blockade, there is no anti-tumor response, and these patients are non-responsive to therapy. However, we believe that we can leverage our CAR T cell expertise to turn these cold tumors hot. That is, increase T cell infiltration in the tumor microenvironment, which will then in turn make them susceptible to checkpoint blockade and lead to tumor regression. Now, in order to devise such a strategy, we need to go to the source of the problem. What exactly makes this tumor microenvironment cold? As you may know, the microenvironment of a solid tumor is a thing of great complexity. It is not simply a solid mass of tumor cells. Instead, it is a heterogeneous mix of a variety of cells and extracellular matrix proteins. And each of these different components play a role in excluding T cells and inhibiting their function, thereby protecting their tumor cells against immune clearance. Decades of research has focused on functionally dissecting the components of this milieu. And from that we know that the key to disrupting this cold immune desert landscape lies within its own cellular complexity. Prominent among these are cancer-associated fibroblasts, or CAFs, primary cells which evolve in the microenvironment during tumor initiation and play a central role in establishment of this immune suppressive milieu. Let me tell you how. First, they secrete extracellular matrix proteins such as collagen, which can form a physical barrier against the entry of T cells into the tumor microenvironment. Second, they secrete a plethora of cytokines and chemokines, such as TGF-beta, which can inhibit the function of the T cells. So it's like CAFs also form a chemical barrier, which protects the tumor cells against the function of immune reactive cells. And third, these cytokines will inhibit immune surveillance by promoting recruitment and differentiation of immune suppressive cells. So you can see how targeting CAFs is pretty much like attacking the heart of the problem and an attractive proposition for retuning a cold microenvironment. CAF targeting is also particularly exciting for us as a strategy for CAR T therapy. CAFs are quite specifically localized to the tumor microenvironment. Plus, they have a very unique set of proteins that are expressed on their surface. So CAR T-mediated targeting should be safe with minimal off-tumor cytotoxicity. A candidate protein on CAF surface that can be targeted thus is FAP, or fibroblast activation protein. In fact, a search of the Human Protein Atlas reveals the widespread expression of FAP protein in the stroma of a multitude of solid tumors, thus indicating that FAP-targeting CAR Ts could have a wide array of applicable cancer indications. In line with our hypothesis, CAF depletion by FAP CAR T cells in mouse models of breast cancer and colorectal cancer was indeed found to boost cytotoxic T cell infiltration into the tumors. And finally, and most importantly, in a small trial, infusion of autologous FAP CAR T cells in mesothelioma patients was found to be safe, with no off-tumor cytotoxicities observed. So, in light of all these facts, we have developed UCART FAP, the first allogeneic CAR T product candidate targeting the solid tumor microenvironment. The objective being, turn a cold tumor hot, make it susceptible to combination checkpoint inhibitor strategies. So, how do we go about doing this? T cells from healthy donors are lentivirally transduced to express the FAP CAR protein. Furthermore, using our superior gene editing TALEN platform, we knock out expression of TCR alpha beta to prevent graft versus host disease, as well as MHC1, with the objective of preventing clearance by patient T cells and thereby increasing their systemic persistence. In vitro cytotoxicity assays, we found that UCART FAP cells efficiently kill CAF cells, which were isolated from patient breast tumor samples, thus validating their activity. However, what is more important to us is assess the advantage of UCART FAP in a physiological context. So we developed an in vitro three-dimensional solid tumor model. Triple-negative breast tumor cells were mixed with patient-derived CAFs and grown in conditions where they spatially recapitulate the organization of these cells in an actual tumor. In these tumor CAF spheres, T cell infiltration is inhibited and tumor targeting CAR activity is hampered, much like in an actual tumor. In this model, we can see how combining UCART FAP with tumor cell targeting CAR T significantly decreases tumor cell survival and increases tumor regression, making a strong case for combining UCART FAP with other T-cell mediated immunotherapies. Overall, here's our vision for UCART FAP as a combination therapy module for solid tumors. UCART FAP infusion post-lymphodepletion in patients will lead to CAF depletion in the tumor stroma. Since CAFs are primary cells with limited regeneration abilities, we anticipate the CAF depleted state to be maintained for a long time. In this scenario, the re-emergence of the patient immune system will be accompanied by increased infiltration of T cells into the now reprogrammed CAF depleted tumor microenvironment, thus turning it hot. These hot immune reactive tumors are now primed to respond to checkpoint blockade immunotherapy. And indeed, they are vulnerable to any other T-cell mediated immunotherapy, including tumor-targeted CAR-Ts or bispecific engagers. So really, with our preclinical project of UCART FAPs, we aim to go for the Achilles heel of solid tumors, declaring open season on these cancers with combination immunotherapies and hopefully getting closer to a cancer-free world. Thank you.
Thank you for joining today. I hope you are as excited as we are for what's coming up next at Selectis. Here's our pipeline. UCART 2022 and UCART Meso will be the next product to enter the clinic. Don't hesitate to join us in the Q&A session for further information.
Nobody can explain to you what Selectus is now capable of. Come see for yourself. Join us tomorrow.
Hello, everyone. This is Simon Harness, Chief Investment Officer at Selectus. We wanted to welcome you back, or welcome you for the first time, to our episode 2 of the Selectus Innovation Days. We're super excited today to speak about new product candidates or development programs that we're planning to bring into the clinic in the next 6 to 12 months. And we have a roundtable of our leading scientists on the phone with us today to answer the questions you've submitted. There are a lot of questions, so thank you in advance for submitting those already, and feel free to submit more questions. And I will start with summarizing a series of questions from different research analysts, including from Citigroup, William Blair, and Kempen, about the UCART 20x22 program, and specifically the series of questions is about are those two targets on the same vector for the CAR integration? And I will hand this over to Laurent Poirot. Laurent, please go ahead.
So thanks, Simon. For the 20x22, the CAR, we're talking about the CAR constructs, right?
Correct.
Yes. So we're not developing a tandem CAR. We're having two CAR on the same bicistronic construct. And this actually is on purpose because having two molecules increase the avidity of the CAR T cell and strengthen the synapse. So this would not happen as well with the tandem CAR. And so we have not measured exactly the density of each molecule, but it's pretty much an equilibrium between the two CARs. Both plays we want a symmetrical role between the 20 CAR and the 22 CARs, so that in case of either of antigen downregulating or being lost, we still maintain strong activity.
Thank you, Laurent. And Beatrice, do you have anything to follow up on that? That was a question for you as well.
I think this, Laurent did a great job, and this was a great question. Thank you for it. I think that it's a great thing that our technology allows us to do, to put the two CARs in the same vector and being delivered in the same cell, because that will help us to deliver both CARs in the same cell, which will give us the opportunity to have a stronger immune synapse, to lead to a stronger killing, and with that potentially achieve really good therapeutic outcomes. So we are very excited.
Thank you, Beatrice. And we just have one small follow-up question on the preclinical data that we have with this CAR, whether there is a significant difference in the dual CAR killing activity versus the single CAR killing activity.
Yeah, actually, this was a very good observation. These data have been generated in vitro, and what we consider is that an activity of lower than 20% is not a significant killing, so we are not concerned by that.
And thank you, Beatrice. A follow-up question on more the trial design and timelines for these new studies to be initiated. So focusing on UCART 20x22, maybe this is a good question for Mark Fortini. What are the similarities or differences in the study structure for UCART 20x22 compared to our currently ongoing trials?
Yeah, so thanks, Simon. So the structure of the studies are very similar, with a dose escalation followed by a dose expansion phase. The one difference that we'll put forward for the 20x22 program, given all the previous experience and data in the non-Hodgkin lymphoma space, is using flat dosing of the UCART cells in this disease.
Thank you, Mark. And maybe pivoting to some of the other programs before maybe coming back to a few questions later on this product. So specifically for Laurent, why did we decide to knock out B2M in UCART MUC1 but not in other product candidates? Like, what's our strategy with the B2M knockout or HLA-E knock-in versus the current lymphodepletion with alemtuzumab and CD52 knockout?
Thanks, Simon. So yeah, this is a very interesting question. So it's definitely not a change of strategy. From the get-go, we have developed two platforms for addressing persistence of allogeneic CAR T cells. So one is basically drug resistance based on CD52, for example, but there are other strategies related to drug persistence. And one is sort of a hiding the CAR T cell from the endogenous immune system. I think both, there's strong values in evaluating both. And so for, simplicity, we've tested alemtuzumab with some product, and we've decided to test the stealth approach with MUC1. And in solid tumor, there's particularly value in trying to see if the endogenous immune system can help consolidating the response. We know the presence of T cells, endogenous T cells in the tumor, is a great prognostic factor for the patient. So having a stealth way of persisting for CAR T cells may allow for endogenous immune system to help actually CAR T cell activity rather than just being here to reject them.
Thank you, Laurent. Very good answer. For pivoting to UCART mesothelin, specific question here. For the UCART meso program, are all three TALEN steps used simultaneously in terms of, like, are those multiplexed edits?
So I'll take that as well. So there's another question a little further that relates to that also in terms of putting multiplex into a process. So we are, because first of all, the electroporation technology is so mild on the cells, we actually like to limit the number of talent at 2 per electroporation. And so we can easily do successive electroporation rather than all combined. The reason why we're limiting at 2 is actually more for regulatory purposes rather than it wouldn't be tolerated by the cells, because the number of potential, it's becoming very complex to list the potential translocation you need to look after, although the level of translocation is very low. On the regulatory aspect, we need to simplify things.
Can I add something? One thing I'd like to add here is like, hi, it's Andre. We also own our own electroporation technology, and that allows us a lot of flexibility on multiplexing one pair of, like double pair of tail ends after double pair of tail ends with our electroporation technologies. And I urge you to come on Thursday to see the way we master this type of technology that Selectus owns since 2010 and have been developing devices in this space. That's the secret behind the scene on how we're multiplexing and we're obtaining such type of spectacular gene editing results in single cells.
Thank you, Andre. Just as a follow-up question to this, this is on a higher level, not related specifically to which gene editing technology you're using, but this is more in general when performing multiple gene edits in one go, correct?
Well, can you say that again, Simon?
Simon, I was speaking while we're talking. Yep. Sorry about that.
I'm sorry. I have a follow-up question, and this is, I think, a follow-up question to Laurent Poirot. If this matters in terms of what technology you're using from a gene editing perspective, or in general for multiplex gene edits, is it cautious to do these separately?
So, I mean, it's a general it's a general consideration. Like it's better on the QC aspects to have more simple, not accumulate too many modifications at the same time. I'm not sure I'm addressing the question correctly though.
Yeah, the screening is very complex. When you go to such a level of high frequency knockouts like this, like multiplexing like this, the regulatory constraints and all the QC constraints is so high that making series of steps makes the thing simplified, because you can wait up to the time the DNA heals afterwards, and then you can bring this to the next steps. And that's all the secret behind the scene. Of course, we're not going to get into all the details because that's part also of the secrets of the company. But that's the healing of the DNA before you get to the next steps, reduces also the regulatory complexity and the QC complexity at the end when you're releasing the product.
Thank you, Andre. A follow-up question on mesothelin from Wells Fargo. Is the mesothelin binder resistant to meso-related peptides when controlled by TCR activation? Laurent?
So there is data out there that's, I mean, mesothelin, part of mesothelin can be secreted, so you could imagine some sort of decoy property that would divert the CAR away from its target. But the CAR we are using is directed against the membrane-bound part of mesothelin. And so, as expected, we have not observed any decoy activity. The CAR is not impacted by those secreted peptides.
Thank you, Laurent. I have a question for the FAP program. Are there any off-target effects in the case of the CAR T cells targeting the FAP antigen that we could anticipate?
Thank you, Simon. That's, I think, the good thing about targeting this particular antigen. As I said, it's very specific to the tumor microenvironment. These fibroblasts normally don't express it. It's only under conditions of tumorigenesis that they start expressing it. So it's pretty localized. And then also, there has been a trial already, limited patients, but the one thing that the biggest takeaway from that trial was that it was very safe. There were no off-tumor cytotoxicities observed. So that's actually very encouraging. So I think we should be good. Thank you.
Thank you, Shubhra. Pivoting back to CD20, CD22, this is a question for Mark Frattini. Can you explain the choice of CD20 to be combined with CD22, considering that some patients acquire resistance to rituximab, and how would that play a role in their potential response?
Thanks, Simon. CD20 is a critically important and proven target in almost all subtypes of non-Hodgkin lymphoma. In addition, CD20-directed therapy continues to be a pillar of therapy in all phases of NHL, not just relapse refractory, hence its importance and to be included here. And yes, while it's true that you can develop resistance to CD20-directed antibodies upon repeated exposure, the mechanism of this resistance is downregulation of CD20 expression. And this downregulation is not complete, and while it may be sufficient to confer resistance to monoclonal antibody therapy, it likely does not confer resistance to CAR T cells, as they are much more sensitive. Also, in addition, with a dual CAR, the addition of CD22 will still provide tumor-specific recognition.
Thank you, Mark. I have a few follow-up questions here. For the question on UCART 20x22, there are currently several CD19 by CD20 bispecific CAR Ts in clinical trials. Could you comment more on the rationale and strategy for positioning CD20 by 22 as a dual CAR versus the CD19 by CD20 bispecific CARs? Is it more a post-CD19 relapse positioning, or would this be completing at the same level with the CD19 CARs? And will it target more NHL patients or ALL patients?
Hi, this is Carrie Brownstein speaking. I think that's a really interesting question, and it goes into all of the details and how complicated and creative, frankly, we all need to be when doing drug development, particularly in spaces where there are lots of options in the clinic. I think for us, and I said this similarly yesterday when we discussed our 22 program, is that it all will go where the data takes us. And 22, similar to 19, is pretty much ubiquitously expressed on B cells in leukemia as well as in lymphomas and NHL. And as is 20 for the NHL piece. I think the data will take us there, and what's nice about the 20x22 program is we're sticking with a really critical, important target, like Dr. Frattini mentioned, which is the CD20 as a pillar in the context of NHL, but bringing in another important target. And I think it gives us an option for whether the data is there or not to be head-to-head with the CD19-directed therapies, or even before CD19-directed therapies, as well as potentially in the context of patients who failed the CD19.
Thank you so much, Carrie. Pivoting away from 20x22, I have a few follow-up questions on UCART Meso and UCART MUC1. For UCART Meso, could you provide more color on your rationale selecting pancreatic cancer as the lead indication, given the higher challenge pancreatic cancer patients represent as an indication?
Thanks, Simon. I think one of the issues with this coming out is to choose malignancies that have consistently high expression of meso as we go into the initial dose escalation. And pancreatic cancer is definitely one of those. And and also given the fact that current therapies for pancreatic cancer are nowhere near optimal. So this provides us the therapy space for that.
Thank you, Mark. One more follow-up question on MUC1. For MUC1, with multiple edits in this program, will the cells contain a suicide switch, given the potency of these CAR T cells and their potential to persist long-term?
So this is not finalized. It could, but there's other pharmacological ways of controlling CAR T cells. So we're evaluating the benefits risk with the benefits risk approach. All the what each edit brings in terms of risk and benefits, and we'll finalize the construct with or without a suicide switch based on this analysis.
Thank you, Laurent. And we're almost out of time, but one important question from Yigal Nochomovitz from Citigroup regarding intellectual property. Where do we stand in terms of intellectual property with respect to the PD-1 knockout and the HLA-E knock-in, given there are a few other companies with similar constructs, either preclinical or in the clinic? Andre, this may be a good question for you.
Yeah, thank you. Thank you, Simon, and thank you, Yigal, for the question. Selectus has been in the gene editing space for 21 years from now, and the knockout of beta-2M or PD-1 and series of other gene editing tweaks has been done before even CRISPR was invented. So it's always very delicate to comment an IP situation, but we feel extremely comfortable currently in the situation where we are. And Selectus has always been a very open company to also promote research and everywhere, while it is still a clinical or preclinical research. Now, none of these products are currently commercialized, so I think that the situation is still in development, and we're always been open for collaborations with third-party companies that would be interested in our IP estate.
Thank you, Andre. And this concludes our time for today. But again, as always, feel free to continue emailing us with questions. You can email me directly at simon.harness@selectus.com with any follow-up questions you have. And we are super excited for episode 3 tomorrow, which will focus on our manufacturing. And we wanted to do this in person for the longest time, but because of the current situation, we're doing everything virtually, and we'll give you a full virtual walkthrough of our manufacturing facilities in North Carolina and Paris. And please join us tomorrow for this very important update on the company. Thank you again, and see you tomorrow.